All right, everyone, we are going to look at Chapter 4, Adaptive Immunity. I want to mention really quickly, though, that I'm going to do this in two parts so that you can look at it, at your leisure, not necessarily all in one sitting. So this is part one of two of the discussion of adaptive immunity. Chapter 4, Adaptive Immunity, is going to be looking at kind of the stages of differentiation of T and B cells, the roles of T and B cells in the adaptive immune response, and then really briefly a discussion of laboratory identification of lymphocytes. Believe it or not, we're going to get through in part one the kind of a review and a discussion about the adaptive immune system and through T cell differentiation, and part two we'll start with B cell differentiation forward. So let's get started. This illustration should look really familiar to all of us. We just completed a review of the innate immune response and realized that it's, it seems relatively simple, but it's, deceptively simple. But it's a very effective external defense for your body. Things like, your skin, the barriers of your skin or chemicals of your body like sweat or lactic acid. And these external defenses of the first line of immunity really not only serve to protect the body from invasion from pathogens, but also creates a really unpleasant and inhospitable environment for any pathogens that may happen to escape the first barriers of the first line of defense. We know the second line of defense is a secondary piece to it. It really is there in case there's a breach to the first line of defense. The host still has a host of non-specific defenses in its arsenal to fight against pathogens. And in this phase, or in this second line of defense, still very non-specific, pathogens can be found in blood and tissues, and they get subject to destruction by, kind of those non-specific actions of phagocytic cells, inflammatory responses like fever, inflammation, the action of natural killer cells, and also those protective proteins that we saw, like acute phase reactants. For the small percentage of pathogens that actually manage to make it past the first and second lines of defense, well, the host has the adaptive immune system. We also call this the specific immune system, acquired immunity, or acquired immune system. And we see it on this illustration reflected as the third line of defense. The adaptive immune system, or acquired immunity, is a series of very complex, targeted immune responses to a specific pathogen. And the good news about this is that we're going to develop some memory to these specific antigens so that when they're encountered again, the host's immune response is much more rapidly deployed and really quite effective. Without a well-functioning adaptive immune response, the host is really highly prone to infections that can be actually quite lethal. We learned in chapter 1 that there are two parts or two components of the adaptive immune system, the humoral and cell-mediated system. Humoral is B cell based, cell-mediated is T cell based. In both cases, both the cell-mediated and humoral, we understand that the adaptive immune response is one where T and B cells recognize foreign material known as antigens, and then the immune system is stimulated to respond in a way that's going to lead to the destruction of the antigen. The response of the humoral and cell-mediated immune system is reliant, first and foremost, on having been activated by some part of the innate immune system. Absent that, the adaptive immune system is not going to respond. Some of the ways that the innate immune system is activated, can activate the adaptive, is through, for example, antigen presentation. We know that presentation by class 1 or class 2 molecules of MHC will trigger lymphocytes, either CD4 or CD8 positive lymphocytes to take a certain action. And we're going to see how both of those cells respond to antigen presentation today. We're going to see that T helper cells, once exposed to that antigen, can take on a number of different responses that will aid the immune system. We saw in chapter 3 the binding of pathogen-specific molecules or pathogen-associated molecular proteins, I guess, PAMPs, on microbes, and how we can see those pathogen-specific markers be recognized by the pathogen recognition receptor of primarily phagocytic cells. And then when we have that recognition by phagocytic cells, a number of actions within the innate system can take place, all of which will lead to activation of the adaptive immune system. The recognition by PRRs of microbial pathogenic markers. Remember, it could be a flagella, or it could be a composition inside of a cell wall. It could be, strands of RNA or DNA. But recognition by these receptors on phagocytic cells can do a couple of things. It can facilitate the uptake or phagocytosis by phagocytic cells. It can target the pathogen for destruction by that complex series of steps in the innate immune system of complement. So it can complement is the serum proteins, the sequential serum proteins that I talked about, like the dominoes, that once you activate it, it leads to cell lysis, among other things. So we can target a pathogen for destruction by complement or phagocytosis. We saw how we can enhance and promote inflammation through cytokine release and chemokine release. And then we can also activate the adaptive immune response. So the PRRs have a number of functionalities within the innate immune system that cross over to activating the adaptive immune system. The host relies on the first and second lines of defense for protection against a pathogen at the first exposure. But the humoral and cell-mediated immune response is going to take time to develop this very effective protection. But it's characterized by not the non-specific mechanisms of the innate, but a very clearly defined specificity for an invading organism. And it's also characterized, the adaptive immune system that is, by its ability to remember. So adaptive immunity is characterized by specificity for an antigen of a pathogen, as well as the ability to remember it. The thing to remember about this, though, is that while it's highly effective after it's been activated, it offers no immediate protection the first time you meet the antigen. That takes a period of time to develop, and when the host is first exposed to a pathogen, you don't be waiting, 5, 8, 10, 14 days for an immune response. So it's really much more effective against the second exposure to the pathogen. It's not to say that there's no benefit to the adaptive immunity, but it really doesn't offer, that kind of outstanding immediate protection that the innate immune system does. So we have two good systems. It's important to note that the adaptive immune system isn't only important in providing recognition of antigens and toxins and things associated with invading pathogens, and then providing that specific response to destroy the pathogen, but it's really important that the adaptive immune system also includes the process of educating, quote unquote, educating T and B cells as they mature so that they will ignore self-antigens of the host. We're going to see as we talk about the maturing or the differentiation of T and B cells that if the cells that are developing either react with self-antigens or don't react with other components properly of the immune system or of the developing cells, those cells are killed by apoptosis. So in general, adaptive immunity, the process of differentiation of cell looks to make sure that we ignore self-antigens. If you remember, if you have a reaction of your immune system with your antigens of yourself, you're developing autoimmune disease. And that's quite damaging and problematic for people. Additionally, adaptive immunity has a goal of conferring lifelong immunity against pathogens. You can't always recognize a titer in someone. And it's true that there are different stages in people's life that their immune system is not as effective anymore. But the immune system of the adaptive immune system has built in that memory for recognition of those pathogens for the life of the host. So let's talk about the cells of the adaptive immune system. As we've been discussing since the first week, there are two types of lymphocytes that are associated with adaptive immunity, T and B cells. They're found in peripheral blood and lymphoid tissues. And a quick note about lymphocytes before we go into some details. First off, lymphocytes are not end cells, and that means that they can further differentiate or they're capable of cell division past the point of when you see them as a mature lymphocyte in the peripheral blood or in the tissues. So first of all, lymph cells are not end cells. They're going to be able to further divide and in fact proliferate, and they do. And lymphocytes have a great capacity to be able to return to the blood from the tissue. So they can be segregated in the tissue and then be called back into the bloodstream. So let's look at the cells individually of T and B of the adaptive immune system, the T and B cells. First, let's talk about why they call T and B cells. Their names are derived from the organs where they develop. T cells are produced in the bone marrow, and then they migrate to the thymus as very immature, young lymphocytes. They develop into mature lymphocytes, and they are then going to involve antigen recognition, that antigen presentation that we see in cell-mediated immune response. B cells, on the other hand, while they're also produced in the bone marrow, they stay in the bone marrow in adults and to develop, and they develop in the liver in fetuses. B cells are involved in the production of antibody, that is humoral immunity. Just going to make a quick note about one thing about the development of B cells. For many, many years, it was hypothesized that there was a location in humans that they called the bursa equivalent. And how that term came up and this theory came up is that researchers discovered that in birds there was an additional site of B cell development, and they call in the birds it was called the bursa of fabricus. Bursa of fabricus. So researchers believed that there had to be some analogous site in the human, and they termed it the bursa equivalent of humans. At this point, from everything I've read, I think people believe that B cells are just primarily produced and developed in the bone marrow, and that the bursa equivalent is not really existent in humans. But I wanted to mention this to you in case it comes up in your readings or, in other discussions around this, that you would understand what the bursa equivalent was talking about. So it was really a hypothesized location of development that didn't really pan out, but you still hear a lot of discussion about it. So when we look at this slide, we've seen the basics of this slide all the way back in the first week. And it reminded us that lymphocytes represent about 20 to 40% of all the circulating white blood cells, and there are three types: B cells and T cells, which we see in the adaptive immune system, and then those natural killer cells that we talked about from chapter 3. Those are involved in the innate immune system, and we're really going to barely talk about it. We're not even going to talk about it in the discussion today about development of lymphocytes. We're really focusing on B and T cells. Non-activated, that is, those that haven't been exposed to antigen yet. The T and B cells that are non-activated look really similar until they get exposed to an antigen, until they're stimulated. Both are small. Both are only just a little bit bigger than a red blood cell. That is, the nucleus of a lymphocyte is generally about the size of a red blood cell. 7 microns or so. Lymphocytes have what we call a high NC ratio, a lot of nucleus to a little bit of cytoplasm. And you can see there's a teeny little rim of cytoplasm here. Both T and B cells are activated by antigen that allows them to proliferate and mature into something we call effector cells. Effector cells are short-lived cells. The reason that they don't live a long time is that effector cells have a purpose. And if they have been activated and there's no reason for them to be there, let's say they were activated by an antigen, but the antigen has been eradicated, we don't keep those cells hanging around forever. We would much rather have the host use the space and the resources of the human body to develop more T helper cells, for example, some naive T cells, so that we have a good supply of naive cells to go and respond to an infection or to a pathogen. So anyway, both B and T cells get activated, they proliferate and they mature into those effector cells. Effector cells are really ones that, an effector cell is really one that's a cell that's responsive to a stimulus and then cause some kind of an action or change. So for example, B effector cells secrete antibodies. B cells get activated and they develop and mature into plasma cells, which are going to secrete antibodies. Effector T cells have a bunch of different functions. The cytotoxic T lymphocyte, once activated, can produce those cytotoxic granules that we've talked about, the perforins and granzymes. T helper cells, once they're activated, we talked about a couple of weeks ago how they can induce memory. They can release cytokines that cause other cells to develop. And in both cases, they can activate the innate immune system. So that's the kind of T cells. Humoral so if we look at T and B cells, and we've seen the cells of the immune system, when we look at the roles of the cells of the immune system, I guess it's important to kind of remember what their function is within the adaptive immune system. So the humoral immune system is based on the premise that B lymphocytes are activated by foreign antigens, exposure to foreign antigens, and then once they are activated, they're called to produce proteins called antibodies or immunoglobulins. The antibodies can be found in circulating blood or other body fluids. And what happens is when they encounter their specific antigen, they bind to the antigen in order to block the pathogen from doing a number of things. So if you remember, I draw a picture of a cell like this with a circle, and then I put a little triangle to represent the antigen. Well, the antibody is a Y, and if we put it upside down, we put the little part of the Y like that, and then the little tail sticks up. That's what antigen-antibody binding is reflecting. We're going to see in the next chapter how that happens, but let's just for now talk about the fact that antigens and antibodies bind. Antibodies bind to their specific antigen that caused their production. So antibodies are specific to antigens in most cases. What does this binding of antibody to antigen cause? What does it do? Well, one of the things that it can do is it can block the antigen's ability to bind to receptors on a host cell and then cause damage to the host. So by covering up that antigen, that little triangle antigen, by covering it up with antibody, that antigen is no longer available. It's covered up by an antibody like a blanket. And that antigen is no longer able to bind to receptors on the host cell and cause damage to the host cell. The other thing that antibody binding to the antigen can cause is it can actually mark the pathogen, the host of that antigen, whether it's a bacteria or a virus. It could be anything. Pollen, as I've said, anything foreign can be an antigen. So it's going to mark that pathogen, the owner of the antigen, for destruction. Mostly it does this by making the pathogen easier for phagocytic cells to ingest and destroy. That process of antigen-antibody binding and making it easier for phagocytic cells to destroy it is called opsonization. And so, opsonization actually has only one eye at the end, not two eyes. I just saw two eyes there. Opsonization. It's not some freakish, I'm just going to try and fix it right now. It's not some kind of freakish spelling. It really is opsonization. And so opsonization just enhances the ability for phagocytes to see the pathogen. And they do it because they can find what did I just do there? They can find the antibody. So that was the humoral immune system, the B cell system, and what happens when antibodies and antigens bind together. T cell immunity, on the other hand, really is referring to the part of the immune system, the part of the adaptive immune system where T cells get activated, again in response to a foreign antigen. I think that you're familiar with both of these mechanisms because we spent so much time talking about it under the MHC complex presentation, MHC1 or MHC2 class antigen presentation. So what happens after T cells get activated? We had a couple of choices. If it's a cytotoxic T lymphocyte or a TC cell, what happens? That the binding with the foreign antigen to the receptor on the T cell will cause a release of granules, perforins and granzymes, that kills the infected host, that has those intracellular, those endogenous pathogens, and that prevents the replication of the pathogen. So that's the first step of cell-mediated immunity within T cells. The second is a little more complex than what we're going to see here, but we're going to talk about it in detail, and that is the actions of when T helper cells are activated. They release cytokines, those chemical messengers that can activate cells in a number of different ways. They can activate other T cells. They can activate B cells. They can activate phagocytic cells. They can, they do all of these things with the purpose of either destroying the invading pathogen or producing memory cells by different methods. So, for example, a T helper cell that releases cytokines that induces B cell production of antibody is designing it to kill the microbe, right? Whereas a T helper cell that releases cytokines that causes inflammation is looking to activate phagocytic cells, again, to kill invading microbes in an innate immune capacity. So we know an awful lot about the adaptive immune system, but to put the pieces together, we really need to look at the different cells and the T cell differentiation and the B cell differentiation, and how, as they're developing and maturing, how is the host's systems there? How is the host able to control the T cells that finally get released into the body and make sure that they're actually functional cells of the immune system? So let's look at the T cells first. T cell differentiation is a 3-week process of maturation in the thymus. In general, those early lymphocytic precursors, we can call them the common lymphoid progenitor, a T cell progenitor, a T lymph progenitor, or precursor. We're going to hear all different terms like that used, but it all means the same thing. Very, very young lymphoid cells. They leave the bone marrow and they enter the thymus through a really fibrous tissue that brings them into the thymus at the corticomedullary junction, the area. Oh my gosh, I have a really sensitive nose here. The junction between the cortex and the medulla. Once these early lymphoid precursors come in, we call them thymocytes. So once they're in the thymus, we call them thymocytes. They're still very, very young. Thymocytes then get called by chemical messengers called chemokines. Remember, a chemokine is just a specialized cytokine. And the chemokine, as we remember, was the one that calls movement of cells. So we're going to have the cells migrate to the outer cortex of the thymus, where we're going to see in a moment some details about how they undergo some gene rearrangement with the purpose of creating a variety of antigen receptors that will be able to detect foreign pathogens, really over the course of the person's lifetime. We want to the gene rearrangement is random, and we're just creating, versions and versions and versions and versions of T cell receptors that there could be. Anything you could imagine, it's going to go looking for. So we're not building specific T cell receptors at this point. We're just kind of randomly building something and hoping it fits to something at another date. T cells that develop to maturity after a bunch of different tests. So we're going to see that as T cells are developing, your body puts them to the test and make sure they're going to function well. Make sure that they're going to be able to see foreign antigens and that they're not going to see and react to self-antigens. So we're going to put these T cells to the test. We're going to challenge them before we let them develop to maturity, where they'll eventually be released from the thymus. They then go out to the secondary organs, the spleen, lymph nodes, things like that, where, if we remember, the spleen is the site of the majority of antigenic contact for bloodborne pathogens, and lymph nodes are the primary site of antigen contact for antigens of pathogens found in the tissues. And that's because the spleen and the lymph node have such high volumes of fluids clearing through them every minute. So If there's going to be antigenic contact, it's going to be in those concentrated spaces of the spleen and the lymph nodes. At this point, it's in the spleen and the lymph node where we're going to see most of the antigenic contact, and that's when the cells are going to differentiate in a post-antigenic way, a very specific way to a specific antigen. Lymphs get segregated within the organs by function, and mainly we're thinking about T cells as being a primary regulatory agency or cell. B cells, antibody production we're thinking of, and then while we're not talking about natural killer cells, we're going to remember just in our head that natural killer cells are involved in the innate and adaptive immune response. So let's get to the stages of differentiation of lymphocytes, T lymphocytes specifically. There are 3 stages: double negative, double positive, and then mature lymphocytes. This is on page, I think 47 of your book. It may help you to have this handy with you, so you might want to put this on pause, take a look for your book, grab this, and look at it from here. I am going to refer back to it, and I did take some screenshots of it, little pieces of it to go through, but you may see me scroll back to this at some point. So as I said before, we have got the thymocyte, or actually the lymphoid precursor or T cell precursor has left the bone marrow and is going to enter in through the corticomedullary junction into the thymus. That T cell precursor, once it's in the thymus, is called a thymocyte. Those early thymocytes are first termed double negative thymocytes, or DN. These double negative thymocytes, or double negative cells, have none of the characteristic markers we expect to see on either T helper cells or cytotoxic T cells. So double negative, they're called double negative because they lack both CD4 and CD8. They're negative for the 2 primary markers for T cells. Chemokines are going to draw the double negative thymocyte to that outer cortex that we saw. We talked about how once they come in through the corticomedullary junction, they move up to the outer core. It's in the outer core that those double negative thymocytes start undergoing what we call random gene rearrangement. And we're just mixing up the genetic coding within every double negative cell that comes in there from the bone marrow. So every single one of them undergoes this random gene arrangement, and they're going to be different. The purpose of this gene rearrangement is to develop diverse T cell receptors for the surface of the cell. And what happens is that when we develop these genes get rearranged and we produce something. Remember, genes, the goal of a gene is to produce something. It's to synthesize something, right? Well, eventually, the cell undergoes gene rearrangement and starts synthesizing some things. They synthesize a very diverse alpha and beta chain. And together, those alpha and beta chains are going to make up what we know as the T cell receptor, that antigen receptor on the surface of a T cell. That's where, when antigen presentation takes place, the peptide is presented to the T cell. The antigen presenting cell presents that peptide, attached into the MHC to a T cell, just like a little gift. And it's that peptide binds into the T cell receptor. So we have to have, innumerable copies of different T cell receptors, because we don't know what's going to come our way. We have to find one that's going to fit. So we're going to develop these diverse alpha and beta chains that are going to make up the T cell receptor. At the same time as we're building those alpha and beta chains within the cell, we're also building 6 other chains. They're not necessarily all different chains. We're going to see in just a minute that we're going to build an epsilon chain, a delta chain, a gamma chain, and a tau chain. And we're going to use combinations of them to form 3 additional pairs of chains. And combined, those 6 chains create what we call the CD3 part of the CD3 T cell receptor complex. So let's take a look at it, and I think this will make sense. So as I said, we were doing that genetic rearrangement to create those diverse antigen binding sites on a T cell. And so here's the alpha chain and here's the beta chain. This area right here is the site of antigen diversity. Very similar to what we're going to see on B cells, on B immunoglobulins. But that genetic rearrangement is what causes this region of the T cell receptor to be variable. Combined it's going to be able to develop receptors against specific antigens. So what happens is we've built the T cell receptor part of our CD3 TCR complex. We've built the TCR. At the same time, I told you we're building other chains. We're building more chains, and we're building this CD3 complex. So we have a pair of chains here, a pair of chains here, and actually an intracellular pair of chains. The first pair of chains, one is an epsilon and one is a delta. The second pair of chains, one is a gamma and an epsilon. And the intracellular ones are called tau-tau, T-A-U, T-A-U. So there's a tau chain and a tau chain, so it's tau-tau. That's a tau-tau pair. So an epsilon gamma, an epsilon gamma, an epsilon delta, and a tau-tau chains. So those 6 other chains are the CD3 part of the complex. The alpha and beta are the TCR complex, part of the complex. Together, they create these 2, 4, 6, 8 chains together, make what we call the CD3 TCR complex. The 6 other chains, the epsilon, delta, gamma, epsilon, and tau-tau chains, are signaling, and those are going to help. These are called signal activation chains, and you can see they build through the cytoplasm of the cell, and they will actually trigger the cell when antigen binds into the T cell receptor, and it's going to trigger this T cell. Imagine this is the cell surface of the T cell. It's going to trigger this T cell to do something specific when it sees this and when the antigen binds there. So that's the purpose of those 6 chains. The development of the beta chain here is actually going to trigger the development of CD4 and CD8 markers on this quote-unquote double negative cell. So we're going to start as soon as we develop this beta chain. We're also going to develop CD4 and CD8 markers, and it becomes classified as a double positive cell. Double positive because I used to have no markers. I had no CD4, no CD8, so it was double negative. Now double positive, not surprisingly, means I have both a CD4 and a CD8, and I have that CD3 TCR complex as well. So I have a double positive cell has CD4, CD8, and a CD3 TCR complex. Double positive thymocytes that express both CD4 and CD8 will continue to rearrange genes for the alpha chain. Double positive thymocytes that once they complete that gene rearrangement, that complex that we talked about forms, that CD3 TCR complex. I want to remind you, some places will call it the CD3 alpha beta complex. they're the same thing, right? We said that the alpha beta chains make the TCR. So this would be, we called it the CD3 TCR complex. You can also call it the CD3 alpha beta complex. They're talking about the same thing. So once we've rearranged the alpha chain, so the beta chain prompted the CD4, CD8 to be developed on the cell surface, became a double positive cell. Double positive cell with the CD3 TCR complex is finalized once the double positive thymocytes have finished their genetic rearrangement of the alpha chain. So now we have a double positive cell with CD4, CD8, CD3 with a TCR complex. That's a nice double positive cell. At this point, this is kind of our first step in making sure that this cell is going to work. We don't want to go out and put all our time and energy into producing a bunch of cells that maybe has a non-functional T cell receptor. So what we do is we go through a process called positive selection. And what that means is that only the cells that can actually have a functional T cell receptor are going to move on. If the cell testing shows that that T cell receptor is not functional, that cell is targeted for apoptosis. It's going to be killed. And it's not going to go through the rest of the testing that we're going to do in development. So if a cell survives this survives this positive selection to show that it has a functional T cell receptor, we say that we end up losing one or the other of these markers. We lose either the CD4 or the CD8, and the cell becomes what we call singly positive. So functional T cell receptors result in a single positive cell. We end up with either a CD8, this is the CD8 marker, this one's the CD4 marker. We call, again, these cells singly positive cells because they have only CD8 or CD4. We're not done though. These cells are not ready to go out into the world. We've tested to make sure that the T cell receptor works through that positive selection, meaning we're only going to take the ones that positively bind in a T cell receptor, that T cell receptor is positively functioning. We're going to do something though. Actually, let me just tell you a little bit about what we're actually testing for in the what else we're testing for here in positive selection. So as I said, we're looking for the functionality of the T cell receptor. What exactly are we looking to see that it can do? We're looking to see that T cells are capable of reacting with the host's MHC. Think about it. If we have T cells that need to bind to a host MHC, if the double positive cell is incapable of binding to a self MHC, that's not a good receptor, right? It's not a good cell, so we want that cell to die. And this testing of self MHC is called the MHC restriction. It's a test to see that the major histocompatibility complex is able to react with the double positive T cell that we're staged at. Similarly, though, we also have to test not just that it's capable of reacting with MHC complex, but that it can functionally react with it. So we've decided that those that don't respond to the MHC complex, they get destroyed. That's easy. If they can't respond to self MHC, boom, they're gone. We also have to check and see the degree to which they'll bind to the MHC. Not too much, not too little. It has to be just right. Goldilocks and the Three Bears. The reactivity between our developing double positive T cell to react with the self-MHC has to be just right. If we have, and we'll say that we're measuring the degree of affinity. I'm going to talk an awful lot about the concept of affinity, I think chapter 9. But what we're talking about affinity, we're talking about kind of the strength of the binding between an antigen and a receptor on a cell, on a T cell, or on any cell actually. So we remember MHCs are not just presenters, they're also what? They're self-antigens. So we're going to see how well these double positive cells react with the self-antigens, and we're going to test the self-antigens of MHCs. If they test too strongly, if they react really, really strongly, then we would worry that there's going to be too much interaction between double positive cells and the MHC complex, maybe on inactivated antigen presenting cells. So we don't want those. We don't want the ones that are overly reactive, too strong of a binding. Similarly, we don't want ones that have a very weak binding, because then maybe they're not going to recognize those antigen presenting cells with MHCs. So we're going to kill those cells off. We want only those ones that have kind of an intermediate, a nice affinity to bind to MHC, to self-MHC, but not so strong that it's too strong, not too weak that it's too weak. Too strong so that it over-recognizes, too weak so that it under-recognizes. So those ones that are on the outliers get killed by apoptosis. The ones that don't respond to self-antigen are killed by apoptosis, and only the ones that bind just right pass through the double positive stage. So as I said, the second step, the second test of the T cells, the second selection process is called the negative selection. Positive selection picked only the ones that were just right. Negative selection, we're looking for ones that are all wrong. Those are the ones that are binding to something we don't want them to bind to. So the second selection process is negative selection. In this case, we're at the point of single positive cells now. Remember, it's only CD4 or CD8. So it's going to test the single positive cell to see if it responds to self-antigens other than the MHC. Remember, we want responsiveness to self-MHC because we have to be able to bind to that and to be able to recognize it and bind to the antigen-presenting cells. We don't want it, though, to bind to, the cells of our, antigens on any cell other than an MHC. For example, we've talked a lot about, because it's an easy one for us to visualize, about blood cells, blood cell antigens. I don't want a bunch of T cells going out and binding off to my blood cells, because all it's going to do is kill them, right? They're not going to bind to my B antigens or my B positive cells. So if we look here, if we have a reaction, a strong reaction binding to a self-antigen other than MHC, remember it's always other than MHC in this example of negative selection, what happens? Cell death. Binding to self-antigen is a bad thing. Kill the cell. If the single positive cell does not bind to self-antigen, and it sees, so this antigen presenting cell can present a self-antigen here. The T cell sees it, but doesn't bind to it, kind of walks away. No binding occurs. That T cell is allowed to exit the thymus and mature. So this process of negative selection and the cell death of cells that react to self-antigen is called clonal deletion. And so what we're saying is that we're going to kill all the clones of the T cell that would possibly react with self-antigen other than MHC. So clonal deletion makes sure that all the clones that come from this one single positive cell is not going to react with self. So we don't want anything escaping the thymus that can react with self-antigen. So if we go back to this stage again, we have at this point, We have those singly positive cells. The double positive cells have undergone the positive selection. The single positive cells have undergone the negative selection, and those that would react here, here we go. Those of them that would react are killed by apoptosis. Those that don't react with self-antigen get to leave the thymus as CD4 or CD8 positive cells, and they go off to mature. So they're mature cells at that point. They're off. They can continue to develop outside of the thymus, and off they go. So to kind of sum up here, survivors of positive and negative selection will express only either CD4 or CD8. We know CD4 are T helper, we know CD8 are cytotoxic. Remarkably, only 1 to 3% of T cells that are produced will survive through negative selection. And just to recap, cell destruction by positive selection happens when there is either a too weak or too strong of a response to self-MHC, or there's no response to self-MHC at all. Negative selection results in cell destruction when there's a response to non-MHC self-antigen. So we want to make sure we can see MHCs effectively. We don't want to respond to self-antigen. That's the two purposes of positive and negative selection. And again, survivors get out. So now we know that T cells have left. They've left as either CD4 or CD8 cells. They're mature cells. They're well developed. They have all their moving parts. And so let's take a look at each of those T cells in a little bit more detail. The first is that we're going to look at are T helper cells. T helper cells, of course, have a CD4 receptor. They recognize the antigen when presented by class II MHC proteins. These are those exogenous antigens, the ones that come in from outside of the cell and brought in. Things like bacteria, right? T helper cells account for about 2/3 of all peripheral T cells, and you're going to see why. There are subsets of T cells that perform many different functions. T helper cells, we know, well, this is obvious, contribute to the immune response in a number of different ways. And as I said, there's 5 different subsets depending on the pathogen that they encounter. And we're going to see an illustration of this in just a minute. Depending on what pathogen is encountered by the antigen presenting cell and what peptide that results in, different cytokines get released that are going to react with the T cells, the naive T helper cells, that are going to help them differentiate into different subsets. So the antigen presenting cells are going to release cytokines that cause those naive, those virgin, exposed T helper cells to differentiate into a couple of different subsets. We're going to look at each of those subsets in detail. There's a T helper 1, T helper 2, a T regulatory subset, and then there's several lesser recognized T helper subsets, a Th17 and Th9. Those are pro-inflammatory T cell subsets, and we'll see those in not as much detail in just a few minutes. But let's look at T helper 1, 2, and T reg cells. T helper 1 cells, their function is to activate cytotoxic lymphocytes and macrophages. And how do they do that? T helper cells, when that naive T helper cell has been sort of bombarded with cytokines from antigen presentation, they develop into one of the T cell subsets. If they're called by the antigen presenting cells, the cytokines, to differentiate into T helper cells, T helper 1 cells, those T helper 1 cells are then going to be able to produce additional cytokines that are going to activate cytotoxic lymphocytes, and it's going to activate some macrophages. And doesn't that make sense that if we have T helper cells that are going to be a little piece of the presentation came from an endogenous pathogen like a virus, we want to activate more cytotoxic lymphocytes, right? We want to activate macrophages that can phagocytize the cells and the debris from the viruses, or if this virus is externally from the cell, just kind of in passing. We see that T helper cells produce interferon. And interferons, you're all too young to remember, but it was a really big thing 10, 15 years ago, where they found out that viruses like hepatitis C could be treated with interferon therapy. It would cause the cells not to be able to replicate, I guess is what I'm trying to say. So if you imagine that T helper cells produce things that are going to ultimately kill virally infected cells, right? T helper 2 cells, their goal is to produce, help B cells. They're going to trigger B cells to produce antibodies, and they're going to regulate some of that B cell activity. They produce interleukins as well. Some of them are going to be redundant. This was an interleukin 2. Interleukin 2 wasn't here, but that's fine. They're a family of interleukins. Often, similar function and kind of redundant function. I don't care so much that you know that it's an interleukin 2. I just want you to understand that an interleukin, an interferon, tumor necrosis factor, those are all cytokines that cause an action. T regulatory cells are characterized not by just having CD4, but they always have CD25 as well. That's what classifies them as a Treg cell. The purpose of these regulatory cells is to suppress the immune response to self-antigen. Well, doesn't that make sense that if the antigen presents self-antigen, we want to have something out there that suppresses the possibility of any immune response to self-antigen. They do this by secreting inhibitory cytokines that inhibits either proliferation of other T cell populations or inhibits the action of antigen presentation. So T regulatory cells are important in suppression of the immune response. So let's take a look at a couple of examples, and we can see the T helper 9 and T helper 17 as well. So let's just follow one or two of the paths and see what's happening. So let's say we have extracellular pathogens or parasites being presented by one of any kinds of antigen presenting cell. So let's just, doesn't matter which one it is at this point, it could be a fungus, bacteria, virus, anything. So let's say this antigen presenting cell presents it to this naive T helper cell. They bind, the antigen presents, the MHC complex brings it up, takes the peptide, presents it to this naive T cell. And at the same time, at this point, the antigen presenting cell knows what it's seeing. It knows the basic concept of what the pathogen is. And it's going to release one of several what we call polarizing cytokines. They're a cytokine that's going to act on this T helper cell. So let's say in the first case, it was a virus. The antigen presenting cell can release an interleukin. It can release some interferons. And it causes this naive T cell to be differentiated by the actions of this cytokine. This T cell is no longer, it's going to differentiate and reproduce. And so what we see if we follow the red line is that that naive T cell differentiates into a T helper 1 cell. And we remember the purpose of T helper 1 cells was to release some more cytokines that it's going to activate cytotoxic T cells and macrophages to influence cell-mediated immunity and kind of the immune phagocytic response. So let's look at another one. If we see one that, for example, we see an antigen presenting cell release, maybe it saw a bacteria. It releases a cytokine, interleukin 4, that again acts on this naive T cell to cause it to develop and differentiate into a T helper 2 cell. This T helper 2 cell is then going to release a number of other cytokines. These are interleukins, and we call these not polarizing cytokines, but effector cytokines. Remember what an effector cell is. It's one that causes another action, right? So the T helper cell, which is now an effector cell, releases cytokines that causes B cells to produce immunoglobulins against the bacteria that the pathogen that was presented all the way at the beginning. Similarly, we can have cytokines released that cause differentiation of these naive T cells to produce T helper 9 cells. Remember, that's a pro-inflammatory one. Maybe it's going to bring mast cells and eosinophils to the area. Or Th17, maybe it differentiates to Th17. Again, that's another pro-inflammatory one. Maybe it's going to activate some macrophages, or again, bring about an inflammatory response. And finally, if antigen-presenting cells release tumor necrosis factor B, it can cause the differentiation of naive T cells into T regulatory cells. And we remember there's going to be further release of that tumor necrosis factor along with some interleukins to suppress the immune system, the immune response. Some cases you can actually have a pro-inflammatory response as well from T regulatory cells. But you can see how the T helper subsets are impacted by these polarizing cytokines released from the antigen presenting cells because of what they're presenting. They know, they're not smart enough to know this is a bacteria, but it knows what it sees maybe on the cell surface. It knows what a piece of the peptide is similar to, and it releases, it's targeted to release certain cytokines that cause differentiation of the naive T cell. That naive T cell was known as an effector cell, so it's going to release specific cytokines, effector cytokines that cause another action. Take a look at this. I know it's a lot of information, but go through it a couple of times. I think it'll make sense, but if it doesn't, you can ask. And that's the end of this. We're going to do part 2 for B cell differentiation in the second part of this lecture. Thanks a lot. Again, if you have any questions, please feel free to contact me.